Well, hello there. This is going to be video 1104, and it corresponds with section 11.4 in your textbook, and we're going to be talking about meiosis. So before we can really get into meiosis, there's a couple of vocabulary words we need to get clear here. The first one is called diploid. So we have cells that are called diploid cells, and we have cells that are called haploid cell. A diploid cell contains two sets of chromosomes. Two sets of chromosomes. An example of a diploid cell would be your body cells. And then on the other hand, we have what's called a haploid cell, and it has one set of chromosomes. An example of a haploid cell would be your gametes. Now, we've talked about this before when we were talking about the construction of a chromosome and when chromosomes replicate. Now, while chromosomes or sister chromatids are attached at the centromere, they are still considered to be one chromosome. When sister chromatids are attached at the centromere, they're considered one chromosome. Now, once they separate, then they're called two chromosomes. So in humans, our body cells have 46 chromosomes, and we get 23 from our mother, 23 from our father. Our gametes have how many chromosomes in them? That's right, they have 23 in them. Now these chromosomes, this 23 pair we have, they're paired chromosomes. One from your mother, or one set from your mother, one set from your father. Now with the way they are paired as homologous chromosomes. So in order to be homologous, first of all, they need to be the same length. The centromere needs to be in the same position. And they should have the same genes that control the same inherited traits. Now they may have different alleles, but they do nonetheless have the same genes. Now meiosis itself has to do with the sexual life cycle. Okay? So you have adults here, and their body goes through meiosis, and that meiosis forms the gametes. Now these gametes are N, meaning what? That's right, that they have a set of chromosomes in each one. They come together at fertilization, and that restores the N back to 2N, which was what would be in the zygote. That zygote eventually will develop, if you remember, the blastocyst, and eventually it will turn into an adult male or female. Okay? Now, you have basically two types of stem cells in your body. You have somatic cells, which are the ones that are going to be your body cells, and then you have germ cells. These are the cells that are going to give rise to gametes. So in my overview of meiosis 1, meiosis itself is a two-stage process. You have meiosis 1, which is the first cell division, and then you have meiosis 2, which would be the second division. Meiosis 1 will reduce the chromosome number by half. So what happens in meiosis 1 is that these homologous chromosomes get separated. Meiosis involves two consecutive cell divisions called meiosis 1 and meiosis 2. So in the phases of meiosis, what exactly occurs during each phase? So in your notes, you're going to need to be writing this down. First of all, you have interphase. Just like in mitosis, meiosis also has interphase, which occurs, and that's when the cell is just kind of doing whatever it does. It's growing. Chromosomes will replicate. Do you remember when the chromosomes replicate? That's right, during synthesis. Okay. And the chromatin condenses, and we're getting ready for meiosis to occur. So in prophase 1, there's a couple of cool things that happen. The first one is that the pairing of homologous chromosomes occur. So in this picture here, the pink one would be the chromosome from your mom, and the blue one would be the chromosome from your dad. Now, when they pair up, they form what's called a tetrad. Tetra means 4, and this tetrad is made up of 4 sister chromatids, or one pair of homologous chromosomes. And just like in mitosis, the nuclear envelope breaks down and the spindle forms. But there's some more stuff that goes on in prophase 1. Well, remember I talked about those tetrads forming? Well, they form during a time that is called synapsis. Now, when these tetrads form, crossing over occurs. Crossing over occurs next to the two closest homologous sister chromatids. So here's our homologous pair. Okay, and as we go through, these two in the middle here, these two sister chromatids, one from the mom, one from the dad, they actually twist upon each other. Twist upon each other. And they exchange chromosomal segments. So if you look at this, you see a little blue on the pink chromosome now and a little pink on the blue chromosome. This is one place where genetic diversity occurs. So crossing over is one place where genetic diversity occurs. Then we go to metaphase 1. And if you remember, this is where they meet, or they meet up in the middle and they line up along the equator. But if you look at this, they're not single file, are they? They're side by side. The homologous chromosomes are. Then we get to anaphase 1, and guess what happens? That's right, the homologous chromosomes separate. So our sister chromatids didn't separate like they do in mitosis. It's the homologous chromosomes separate, and they pull away from each other, or they pull apart in anaphase 1. Telophase 1, that spindle breaks down, the chromosomes uncoil, and your nuclei reform. And then cytokinesis will follow telophase 1, and the cell divides. Now, at this point here, we are no longer 2N. Now we are 1N, or N. And after cytokinesis, it doesn't go to interphase. It goes straight to prophase 2. Prophase 2 is going to be the second set of phases that begin. That spindle reforms, and the chromosomes condense again. They meet in the middle. Now this time it's the haploid number. Before it was remember it was the homologous chromosomes. Now these should kind of look just like they did in mitosis, except you have two of the cells doing this. They line up at the equator. They go into anaphase 2, where the sister chromatids are pulled apart. Sister chromatids are pulled apart. Both of those cells then pinch in the middle. Oops, sorry. And they reach the poles, the nuclear membranes, and the nuclei reform. They go through cytokinesis, and now we have 4 haploid cells. Remember, haploid means that they're going to have N number of chromosomes, or half the number. So why is meiosis important? Well, first of all, it consists of 2 sets of divisions, and it produces 4 haploid daughter cells that are not identical, and it results in genetic variation. Genetic variation meaning that depending on which sperm and which egg fertilize or get fertilized is what the offspring will look like. So there's 2 places where this genetic variation can occur. One is it depends on how they line up at the equator. 4 gametes with 4 different combinations can result, so depending on how they get split apart. And then you also have genetic variation during crossing over. Okay, so when they line up, that's going to vary, and then when they cross over, that's going to cause variation. And then fertilization is another type of genetic variation because it depends on which sperm and which egg form the zygote. In male, remember, gametes are called sperm. In plants, pollen grains are what contain the sperm cells. In females, generally only one of the cells is produced by meiosis. The female gamete is called an egg, and an egg cell in some plants. The fusion of the male and female gametes is called a zygote. This zygote is what undergoes cell division by mitosis, and it eventually forms a new organism. So let's look at the differences between mitosis and meiosis. So mitosis, they're genetically identical. Meiosis, they're genetically different. Mitosis produces diploid cells. Meiosis produces haploid cells. Mitosis would be a type of asexual reproduction. You get your body cells. And then meiosis is the early step in sexual reproduction, and you get gametes. And then mitosis has one cell division, and meiosis has two. I know that says once. That should say one cell division. Oops. Sorry about that. That should say one cell division. There you go. Now, what happens if meiosis doesn't happen correctly? What can happen is a process called non-disjunction. That's where they fail to separate. So this can cause a monosomy, or it can cause what's called trisomy, where monosomy is where you're missing a chromosome in one of the gametes, or trisomy is where you end up with an extra chromosome. So if you take a look at this picture I have here on the left-hand side, so this is where the non-disjunction, they didn't separate like they were supposed to. And when do they separate? That's right, anaphase. So what happens when they didn't separate correctly, instead of having 2 and 2, so these two, one of the large and the small one should have been over here, and this one should have one of the large. Instead, what ended up happening was 3 of the chromosomes went over there and only 1 went here. So when they divide, you end up with 1 and 1 in there, and then 3 and 3 in this one. So if non-disjunction occurs in meiosis 1, all 4 of the gametes have the wrong chromosome number. If it says +1, that's trisomy. -1 would be monosomy. You might want to stop the video here so you can take a look at that. Now, if non-disjunction occurs in meiosis 2, that's not as detrimental because you had the 2 in this one and the 2 in this one. It's just that this one here did not separate. And so what that causes is you have an extra one, or trisomy, in one egg or sperm cell. You're missing one in the other egg or sperm cell, and then you have the correct number in these last two. So with non-disjunction occurring in meiosis 2, it's not as bad as if it occurs in meiosis 1. Here's another way of looking at this. And that ends this video.